Has started.
Well, good afternoon, everyone. Thank you very much for joining in on our latest edition of the CellCan webinar series, Next Stop Vancouver. Today's presentation is from Dr. Jamie Piret, the technology development tailored to address cell therapy manufacturing challenges. Before we begin, I'd like to say a few words about CellCan for those of you that aren't quite familiar with our organization. Just to remind everyone what we're all about. So CellCan was incorporated in 2014 as a knowledge mobilization not-for-profit focused around the niche of cell and gene therapy manufacturing. Our vision is that cell and gene therapy manufacturing must operate under a common seal of quality with a vision that optimal manufacturing practices in Canada must take place to increase the quality, safety, and feasibility of CGT within our ecosystem. We have four main business priorities within CellCan: to build a strong Canadian network, and this involves a lot of strong collaboration, which helps break down the silos and share knowledge around best practices within our very important manufacturing niche. To advance regulatory standards, we work together with our network partners and various stakeholders around the country and with our excellent regulatory authorities at Health Canada to push forth different policy changes and as well to help craft the regulatory reform that's taking place within our country right now. We generate effective outreach through mechanisms such as this webinar series right now, all with an effort to position Canada as a world leader within cell and gene therapy. If you haven't heard already, we're very pleased to announce that our Advanced Training Institute has launched. This is focused around delivering GMP-specific skills to HQP all across the country and indeed globally. You can find more information on our website, cellcan.com, as well as by going to ATMP training, as in Advanced Therapy Medicinal Product training.com. We have two main learning paths right now, and essentials of CGT GMP manufacturing, which is sort of an executive level type of under an hour learning path, and as well our very robust hands-on GMP virtual hands-on workshop, which is sort of the online virtual interactive component that has really vaulted forth after our in-person GMP hands-on workshop that's taken place over the last several years here in Montreal at the CTC. To give you a very brief overview, our interactive modules are designed to be to work directly with the participant to test knowledge at various time points and to learn from actually seeing the visualized representation of all the concepts and theory that are being delivered, and as well as to make sure that the knowledge is taken up within defined checkpoints, including interactive learning modules like the one you see here on bioreactors. We're very pleased to say that Canada's next generation manufacturing supercluster engine has recognized our training program. You can find out more information on this at Engine's website. They have an AMP Up program where you may be eligible for up to 50% of the training costs covered for employees of Engine member companies. So please check out engine.ca/upskilling for more information. And without further ado, today I'd like to introduce Dr. Jamie Piret. He's certainly a key member of the CellCan network, and he's here to talk about technology development tailored to address cell therapy challenges. Dr. Piret is a professor in the Department of Chemical and Biological Engineering and the Michael Smith Laboratories at UBC. After completing his bachelor's degree at Harvard, Jamie went on to complete his doctoral studies in chemical engineering at MIT, after which he went to UBC, where his research is focused on the R&D of innovative process and device technology for mammalian cell culture and therapeutic protein or cell production. Jamie's multidisciplinary research ranges from bioreactor engineering to molecular biology, often including collaborative work with scientists and industry. Dr. Piret was the founding bioengineering leader of the Canadian Stem Cell Network and with our very own Regenerative Medicine and Cell Therapy Network here at CellCan. So without further ado, Jamie, I'd like to turn it over to you. Welcome. We really look forward to your talk.
So thank you very much, Craig. I expect
if I can just mention, right now we see a presenter view. Excellent. So as Vanessa's just mentioned in the chat here before we get started as well, on the top right, if you hover over the top right of your screen, you'll see people and chat. So please don't hesitate to interact with us. Type any questions you may have within this chat box, and we'll do our best to have them addressed by the end of the presentation, if not sooner. Again, over to you, Jamie.
Okay, super. Thanks for all your support over the years, really, and it's been a pleasure to be part of this network that I think is really addressing a key need for the future of Canada, for the future of the Canadians in this promising new area. As you suggested, my lab and my training actually originally was oriented towards recombinant therapeutic protein manufacturing. And I remember, well, first of all, I came to UBC in that area, but within a couple of years was very fortunate to start working with Connie Eves on hematopoietic stem cell expansion and culture. And so got involved in this field very early. At a time where my colleagues who had gone in particular to biotechnology companies to produce recombinant proteins most often expressed a lot of skepticism as to whether or not I might be able to do research on it, but whether or not we would really get to the point of manufacturing cells for therapy. And they had a good reason for that. I mean, those cells are so much more complex than monoclonal antibodies, for instance. And yet they and those companies were struggling to get consistent monoclonal production, such a complex product. How would it be possible to achieve the same thing with cells for therapy that are so much more complex? And so one answer that I always had was, well, cells are used for therapy already. Hematopoietic stem cells for transplantation, something like 50,000 cases per year, hundreds of millions of blood units transfused, hundreds of thousands of organs. So really cells are used for therapy. But they, of course, had a good point, because this new wave of cell therapy manufacturing really relates to the blue lines in the schematic, where in fact the cells are being changed by the manufacturing process. They're not minimally manipulated in ways that the established therapies are. These new therapies are really changed by the manufacturing process. It can be weeks that embryonic stem cells generate cells that are beta cells that can be used to treat diabetes, for instance, or they may be genetically engineered, as is the case for CAR T cell therapies. And so, of course, that is a big part of the challenges that we face in moving forward with this technology. And we must do so because, for instance, in CAR T cell therapy, we've extended the life, or the field in general, of course, has extended the life of I think an 8-year-old by 10 years by now, maybe a lifetime with a single CAR T cell treatment. And so it is possible, and it can be remarkably effective. Something on the order of 50% of those CAR T cell therapy clinical trial patients went into remission on average with the basically the cancer cells disappearing within, I think, 3 weeks in the case of the Novartis trial. But really the challenge now comes to the bioprocess engineering to some degree in order to be able to do this robustly and reproducibly. And so my research is very much in that area. I work in areas related to cell culture optimization, both for recombinant protein production, but also in areas related to regulatory T cells that I'll present today. We work on genetic engineering of the cells in both I've worked on microfluidic stem cell culture and analysis, and indeed a big success of my Michael Smith labs is our contributions to the formation of Cellular, a company that was formed in 2012, but just a few months ago had the largest biotechnology IPO in Canadian biotech history, and has a market cap that is larger than a biotechnology company in Canada. And so it's really exciting for us to see that sector developing. I'll also present, but I'll present today on the devices for cell therapy manufacturing. I work on immunoisolation for diabetes therapy, but also an acoustic closed system for cell processing, as well as Raman spectroscopy as a process analytical technology. And I'll present on those two latter as well. Of course, the immune cell therapy is a big excitement and advance these days, in particular since the 2017 approval of CAR T-cell therapies that Novartis and Kite advanced. And I like this slide that was put together to show how immunotherapy clinical trials initiated since 20 years ago have really been building up to this moment. And there's been so much effort going into actually using these cells that are losing a battle against cancer to recover them from the patient, activate them, genetically engineer them to bind cancer cells, grow them up in culture, and then reinfuse them into the patient to be so effective. And so that has become a very important area for the sort of bioprocess engineering that we're involved in. And I've been fortunate at UBC to work with Megan Lemmings, who along with Laurie West developed a regulatory T-cell approach to immunotherapy that could be used to prevent transplant rejection, graft versus host disease, or organ transplantation. One could suppress the rejection by using regulatory T cells. And what Megan and her collaborators developed was they could recover regulatory T cells from pediatric thymus as a byproduct of cardiac surgery for children, where essentially the thymus is in the way of the cardiac surgeon in such a way that a large part of it, if not all of it, can be removed in order to enable the cardiac surgery. And so these cells, this is a rich source of thymic Tregs that through culture and through expansion can be obtained in sufficient numbers to transplant patients for something like graft versus host disease. An important attribute of this approach is that the cells, the naive T regulatory cells, are pretty much the only ones in the thymus that have a high level of CD25. And so by a simpler magnetic selection, those cells can be recovered without the need to use flow cytometry, which is often the case for regulatory T cells that are recovered from the blood. And so that's an important attribute when one wants to move into GMP manufacturing. To be able to do magnetic bead selection is far preferable to anything involving flow cytometry. So another advantage of these pediatric thymus cells is that they are capable of expansion in vitro. First of all, as is shown here, the red versus blood Tregs in blue. But then also you see here by the length of the telomeres, you have a potentially greater potential for expansion for these cells, as well as this being a rich source of cells. And so along with Katie McDonald, who's a bioprocess engineer working on this project, we have worked on the classic sort of development of a process for clinical trials, where the challenge was to move to reagents and methods that were suitable for a GMP environment, such as eliminating the need for antigen-presenting cells and replacing that with products such as the Dynabeads or stem cell technologies has a soluble activator that we readily separated without cell losses, as does Miltenyi as a T-cell transact. And so these kinds of experiments are what we did, where you can see higher expansions, for instance, with this stem cell product and the Miltenyi product, or no, those are the highest. And here, actually, I'm so sorry. This is the cell presentation, and so levels quite effectively while removing the requirement for cell activators. However, you see here the quality of the cells, where FOXP3 is an indication of regulatory T cells. And then here's a functional assay where we use regulatory T cells and see how much they can suppress the expansion of cells from PBMCs. And thereby, one can see that essentially it gave us the highest performance, and we intend to move forward with those. But interestingly, the other alternatives, the other non-cellular activators, actually were quite effective in this. And so it might, in fact, only be a matter of further optimization to match the Dynabeads. And that's actually something that we're interested to do, especially since Stem Cell is a company in the Vancouver area. Likewise, we analyzed effect of regulatory T-cell expansion of culture media. And so comparing with the Dynabeads, Immunocult, ex vivo, as well as Optimizer, in this case, it was the stem cell product that gave us the highest fold expansions, as well as high quality cells and the expected suppressive activity that we needed. What was a surprising sort of result in this was that cryopreservation timing was a critical process parameter, one that we had not expected but had a big impact on the quality of the product, where what we did is analyze cells that were activated at time 0, re-stimulated at day 7, and then cryopreserved either at day 14 or 15, or they were re-stimulated and cryopreserved later. And so that's what these plots here summarize. And what you can see is that the viability of these cells here were decreased after thaw and overnight culture. Indeed, even more were apoptotic. And here you can see an interesting phenomenon where the quality of the cells from the point of view of apoptosis, as well as FOXP3 suppression, was quite high for a couple days after re-stimulation, so after day 14, but not at 5 days and at later time points. And so this showed us that really the timing of this cryopreservation relative to the re-stimulation was a critical process parameter. And that is what one seeks in developing bioprocesses of this type. There's variables among so many that have this kind of impact. And so this title of the slide pretty much ended up being the title of our paper, which I cited in the first one of these slides. It's McDonald et al., 2019. And so here I like to show how, that thymic Treg expansion process can seem simple to some degree. There are a few variables that relate. However, it actually is a very complex process where there are so many variables, including the donor cells, the cell concentrations that we choose to use, the viabilities we obtain, the phenotypes of the cells, all of this dynamic, and over time, the medium composition, pH, O2, CO2, nutrients in the medium, metabolites that accumulate. And so, in fact, we have a lot of potential critical process parameters, kind of quality by design that the whole field is moving towards, and CellCan can advance as a network of facilities doing manufacturing, is really in the identification and sharing of knowledge about the critical process parameters that are identified in the varied processes that we are addressing and encountering, and thereby not only maximize the yields, but also understand these systems in a way that, from a rare point of view, would give confidence to regulatory authorities that we have a good understanding and thereby control of our process performance. So one thing that's important to recognize is that cell therapies are regulated as biologicals. So biologicals, as opposed to drugs, are these much more complex products that cannot be fully purified or fully characterized. Monoclonal antibodies are biological, but far simpler ones. Vaccines, especially, for instance, the AstraZeneca one, is a complex biological where not only is each individual viral or particle complex, we have a population of them, and they can't be purified. And so because these products can't be fully purified and characterized as drugs are, both the product, so the kind of analysis that I was just showing, need to be validated, but also the process, because the recognition is that if a product, if we cannot identify that the product has been altered during the process in a way by the release criteria, we might and often could detect variations in how the process performed, how fast the cells were growing, how much they produced a desired. And so I like to quote here something that Scott Gottlieb said a few years ago as commissioner, when he was commissioner of the FDA, where he talks about, in contrast to traditional drug reviews, where 80% of the review is focused on the clinical portion of the file, the clinical results on the humans that were tested, and about 20% of it is focused on how the material was produced in the first place, the quality of the product. And this would apply to monoclonal antibodies as well. He'd say, in contrast to that, for cell therapies, this general principle is almost completely inverted when it comes to cell and gene therapy. The clinical efficacy is often initially established early and sometimes in a small series of patients with very clear benefits, as has been the case for CAR T cell therapy trials. But it is the product questions that are much more complex and uncertain. And so for this reason, it is crucial to focus on these sorts of aspects in order to maintain robust and effective manufacturing processes. So Janet Woodcock actually contributed to something that's referred to as quality by design in manufacturing, where essentially it was defined as a means that product and process performance characteristics are scientifically designed to meet specific objectives, not merely empirically derived. We produce it the same way every time, and so we get the same apparent quality, but really to understand when there are variations in how the production process performs, when are those within ranges that are acceptable or not, and ideally what mechanisms are responsible for this in such a way that in an unexpected situation one would be able to analyze and understand the quality of the product. So really, so quality, of course, is good. Represents an acceptably low risk of failing to achieve the desired clinical attributes, and there are many international efforts to develop the sorts of standards associated with this, including working with the design space. And I'm very interested in my research to flesh this out and apply this in the real context that we deal with, including through linkages and collaborations with members of CellCan, such as Greg Corbett in Alberta and Denis Montreal. So essentially, that's the end of the culture optimization part. And so now I'll also address that we're really in a window of opportunity for cell therapy processing devices. With so many clinical trials, varied therapies, there's a need for this safe and effective production that need to be economical, some industrialized, some hospital-based. And so there's really a window of opportunity that includes relatively small scales. In the recombinant protein area, moving to manufacturing means moving to 1,000, 10,000 liter tanks, whereas especially for autologous therapies and hospital-based therapies, one is often dealing with just for instance, cell concentration and washing at a 1 liter of medium scale. As well, an important area relates to process technologies that are after the cell concentration and washing. So here, this is a schematic of a CAR T-cell therapy process where essentially there are many stages: purifying the cells, activating the cells, transducing them, expanding them, separating them, purifying them, and at the most common unit operation in this is the cell separation, and what's referred to as washing. So when moving from one medium type to another. And so that is something that effectively is addressed currently by instrumentation that's essentially derived from the blood processing field. And that makes sense because often single units of blood are being processed. Used to be referred to as CPACs, has been put together as the Cephia continuous centrifuge system, essentially a cylindrical centrifuge that allows the cells to be centrifuged in a closed system. And so this can effectively accomplish the sort of cell concentration and washing that's required for CAR T-cell processing. But it really involves cell stresses and old surface cell losses that in the blood field are of no great consequence. A 10, 20% loss of the cells is just a cost of doing business in some sense. But in cell therapy and manufacturing, those might be the only cells that the patient has, for instance, to provide to enable a cell cell therapy that could save their lives. And so there's also an incentive to tailor these measuring devices, either incrementally, as has been the case for that centrifuge system that's been adapted to this application, or disruptively, really adopting new technologies and new approaches that are better tailored for the specific needs of this new field. And so one area that we've been involved in, and I'm involved in a company called Sonocept that developed an acoustic cell separation device back in the 90s that's been commercialized for over 20 years now by Applicon in the area of protein manufacturing to retain cells in what's referred to as a perfusion reactor. And so essentially we've adapted that to this new situation where we can concentrate the cells in order to wash them in ways that are more effective than smaller volume separation steps like the one for CAR T-cell therapy. So as an example of that, for the centrifuge system, one might start with 50 mils of cells, dilute them tenfold, and then concentrate them back down to 50 mils, and then dilute them tenfold and concentrate that down to tenfold to have a 99% washing, a 99% removal of the soluble components, maybe DMSO from a cryopreservation medium. Whereas a device here, we can concentrate the cells and in a much smaller volume, wash them with basically perfusing medium through them. And so instead of using something like 1 liter or 900 mLs for a 99% wash, we can actually use 50 mLs for a 99.99% wash. So a even greater clearance of substances that are not desired in the next step in the process. And so not only shows how we can have a higher quality washing from that point of view, but also we can reduce the use of reagents that would be costly in this setting. But even more important than that, because it's such a small volume, instead of using a separate and separately validated wash solution at this step, one could actually use 50 mils of the medium that's used in the next step, losing a little bit, but essentially eliminating an input to the process. So that's good. You want to simplify your process. You want to reduce the number of materials that go into your process. That improves reliability and safety issues in a way that is tailored to fit the needs of this kind of process. From an engineering point of view, there are a lot of interesting dimensions to this. A feature of the acoustic system is the energy that's put in the cells in this acoustic system actually dissipates ultimately as heat. And so there's an engineering need to address that and to operate it in ranges where the temperature might not vary more than 2, 3, or maybe as much as 5 degrees. And so what we do there is essentially operate the device with air cooling. And this is actually an illustration of computational fluid dynamics go into determining the flow distributions inside the device as well as the temperature distributions so as to provide the assurance that the kind of exposures of temperatures are ones that would not lead to a negative impact on the cells. So that's the kind of process and device understanding that, of course, is important in this sort of regulatory setting. So the other example that I wanted to give was with respect to Raman spectroscopy and the need for process analytical technologies. And so I'll just step back here and show you the kind of contrast between the field that I come from and the field that we're involved in, where, as I said earlier, protein biologics, very large, much larger scale, even allogeneic manufacturing in the cell biologics area is often on the scale leaders, not much more, although we can hope that that will scale up more in the future. There are issues with immunogenicity, especially with aggregated monoclonal antibodies, for instance. Immune rejection, much bigger challenge, and indeed the immunoisolation that I'm involved in is seeking to address issues related to that for diabetes therapy. Protein biologics are generated essentially from monocultures of cells that produce the proteins, whereas we're dealing with mixed cell populations, not only mixed cell population, but cells that differentiate from one cell type to another often, or from naive to exhausted cells ultimately in the T cell case. And there can be some adverse effects of protein biologics. You just have to listen to a television advert to realize how many they can be. But cellular biologics have these risks to address. And I think most of all, this complexity of the product is what's remarkable to me. And I like to illustrate that in this way, where essentially the area that I started out in producing monoclonal antibodies is something like sending a cruise missile, to try to win a war. Sophisticated, highly targeted, very complex in its own right. But when it comes to cell therapy, we're essentially sending in the battleship. We're sending in a device or an entity that can produce things like antibodies, but also has many other weapons, and also is far more complex than monoclonal antibodies. So this, for me, also is an opportunity for innovation in area. And in particular, because the process should be validated, it is important to develop process analytical technologies that can monitor how the process is evolving when each lot is being produced. And a challenge in our field that I like to illustrate with this figure here is that our work is most often based on infrequent analysis. CAR T cell therapy manufacturing probably sampled once or twice over 10 days or something of that order. And also lagging phenotypic markers. This is a figure from a publication of ours where we showed that, in this case for murine embryonic stem cells, the phenotypic markers, SSEA1 and OCT4, drop off very slowly, dropping down really only around 5 days after removal in this case, whereas the functional markers, which are the important ones for the therapy, of course, drop off much faster. Even colony-forming cells, embryoid body, as well as chimera are really something that drops off very fast. And so it's not only important to develop improved means to find the biomarkers of these kinds of functional consequences, but also ideally one would want to be able to sample virtually continuously without disturbing the culture. And so that's why I've been very interested in Raman spectroscopy in this context, where essentially Raman spectroscopy is based on the fact that if you shine a laser on a sample, almost all of the photons will be scattered at the same wavelength, but a very few, 1 out of 100 million, something on that order, will be scattered wavelength, leaving behind energy in a chemical bond that it interacted with. And the Raman shift that results from that change in energy is characteristic of the chemical bonds that it interacted with, such that, for instance, here for benzene, you have this Raman shift with a very prominent peak at 1000. And so this is pure benzene. That's a pretty simple Raman. Whereas you might guess that cells will have a far more complex Raman spectrum, and indeed they do. As is illustrated here, for even macromolecules within cells, they have quite complex spectra, where you can see for the proteins, we have this peak here that corresponds to the 1000 that you saw with the benzene, and that essentially results from the side chain phenylalanine. And so this peak here allows us to distinguish in a complex spectrum the content of proteins, as you can see, histones, collagen, as well as actin, and thereby other peaks that are actually quite a bit more prominent. All of these are normalized to each other for lipids, as well as glycogen, which varies a lot in embryonic stem cells, and also acids, RNA and DNA. And so essentially what we can do, and the work that we have performed, is using a Raman spectroscopy system in a microscopic system that essentially allows us to analyze cells even at a subcellular level. But what we are doing is discovering the Raman shifts that could be indicative of bioprocess monitoring variables. So the advantage of spectroscopy is it's reagentless. It's actually a characteristic of the substance, so label-free, non-descriptive measurement of chemical composition. So in most cases, the macromolecular composition of the cells, the proteins versus nucleic acids. It's information-rich, non-invasive, and other alternatives of that type would be light absorbance that has much less information in it, infrared that has a lot more interference from water, and NMR would be another one, but that requires much larger samples. And so we've applied this in a number of contexts, including here I show how we analyze cells that were undergoing apoptosis induced by camptothecin. And you can see in these image sequence of images that we have an exon 5 pi negative double CHO cells in this case that up around 33 and a half hours start to exhibit an exon 5 and then later pi as they get into secondary necrosis. And so what we did was analyze by Raman spectroscopy the composition of these cells. And so in this spectrum here, you see variations between uninduced 24-hour camptothecin as well as those that begin to be an exon 5 positive. And you can see that essentially there's a big drop, for instance, in nucleic acids, as you might expect, given the consequences of apoptosis in breaking down DNA. And in addition, very substantially RNA, as can be distinguished by this peak. And so essentially the Raman spectroscopy allowed us to identify the macromolecules that were happening, and then we could go back to biochemical analysis and show that in particular the RNA was significantly different on the onset of apoptosis. I think maybe the most remarkable thing here is that we're actually detecting these changes even in the annexin 5 negative cells. And so those are cells, that's the blue lines here. Those are cells that would not be detected by their early apoptosis marker that we normally use in flow cytometry. So really showing how we can get a timely result from the point of view of this. Any kind of bioprocessing, we would be concerned to know when there is an onset of apoptosis. That would be an indication that there may be a need for an intervention, maybe addition of nutrients, or maybe towards the end of the process, this would be the time to harvest. We think this really fits nicely into the area related to cell therapy manufacturing in ways that will be valuable as this technology advances. Here's just another image. It shows from a collection of samples that we actually could distinguish primary necrosis from secondary necrosis that comes after apoptosis, whereas viable cells or autophagic cells were not distinguishable by their composition. And so really now we can get all kinds of insights into the cells. And indeed, we've explored these kinds of things over the years as an analytic for a well-controlled manufacturing process that would have an expected evolution in its how the cells appear. So first, analyzing differentiation of embryonic stem cells, autophagy induction, as well as the cell death modes that I just showed you. Publications, many more than this, related to the data analytics, because the analytics of processing the data from Raman spectroscopy are most often done in a manual way, and in a clinical setting that would have to advance to a more unbiased automated approach. We've explored visually offset Raman spectroscopy that can be used to analyze cells that would be, for instance, in a cell culture bag, and so non-invasively. And then also related to insulin in the last few slides of this talk. So the last topic I wanted to address was related to Raman spectroscopy, but in the context of this promising 7-stage cell manufacturing process that Tim Kiefer at UBC published in collaboration with Betalogics. And essentially, this is one of the world-leading protocols. So I'm fortunate to be able to work with, in collaboration with him, and in analyzing cells as they go through a differentiation process towards beta cells that would be used in cell therapy for diabetes. In fact, ViaCyte has cells that they are testing now. They've actually recently started a phase 2 clinical trial with their implantation devices of a similar type. So analyzing cells at various stages, the Raman spectroscopy starts to distinguish them most effectively from stages 4 to 7. And so you can see an evolution in these profiles, where essentially here you have higher and higher levels of what actually turns out to be disulfide bonds, as well variations in lipid levels. All of these, I should say, are normal. The nucleic acid content peak at 780. And so that allows us to normalize the data so as to make up for when there are different quantities of cells exposed to the laser and Raman spectroscopy analysis. And so this kind of evolution in the profiles and the macromolecular composition of the cells is something that is characteristic. And since here in a set of half dozen trials and just PCA analysis of the Raman spectroscopy, you can see one of those experiments that had failed to produce the appropriate beta cells was distinguishable by this principal component analysis as trial 3. That's good, but one really seeks to have more of an understanding and a mechanistic understanding of what is being analyzed in order to give confidence that in some new situation, this would remain a device that could give a reliable response. And so a remarkable feature of working with cells that generate insulin and glucagon through this differentiation process is that insulin has a prominent, this is just purified insulin, has a prominent disulfide peak because it had a high content of disulfide bonds compared to most any other protein. But in particular, most cellular proteins have no disulfide bonds. And so that is something that when cells accumulate insulin, they accumulate disulfide bonds. Likewise, glucagon is a big part, is what's produced by alpha cells, and so a byproduct in this process. And they have a high tryptophan, which is has a strong Raman peak at this point. So essentially, showing you the same profile that I showed you before, this increase in the disulfide bonds that we were analyzing actually correlates with insulin accumulation, as well as this increase in the tryptophan peak correlates with the accumulation of glucagon that is also stored intracellularly in such a way that, for instance, here showing a comparison of insulin messenger RNA going up at stage 5, 6, and 7. Likewise, at stage 5 to 6 and 7, you see an increase in this disulfide bond peak. And we see similar correlations with direct measurements of insulin, as well as here you see for glucagon the same kind of phenomenon. So this is great, of course, because in some sense, the insulin is the drug substance. The cells, of course, are what's implanted, but their ability to synthesize and accumulate insulin is an indication of their potency. And potency assays are difficult to have, and to have that potential in an online manner is remarkable. So essentially, overall, we see Raman spectroscopy as an analytical approach that could monitor the process as well as the product, potentially to find biomarkers for clinical outcomes where we think insulin, of course, is a good candidate. And so on the one hand, analyzing during the process to see that each of these peaks, and we normally look at ratios of peaks, evolves within the expected range, is part of what would be in the process analytical technology end product characterization. So analyzing, for instance, for the insulin, as is indicated here. Okay, so essentially I tried to outline technologies for cell therapy manufacturing and the kinds of advances that are happening, and these involve collaborations across Canada one way or another. The quality by design cell therapy manufacturing that I showed you was related to the work with Megan Levings, but a lot. The whole group collaborates with groups in Edmonton. We expect to move a clinical trial into the ACTM in Edmonton, the cell therapy manufacturing facility there. We've already done some testing with them. The cell concentration and washing system, acoustic cell processing, we're involved with this local company. That, by the way, Millipore is developing a similar product. And then embryonic stem cell to pancreatic progenitor label-free monitoring is something that we're working on developing a non-invasive online cell differentiation system. But then also that's involved in really national collaborations, one way or another, most often led by Tim Kiefer. And so finally, I appreciate your attention and be happy to answer any questions. I've tried to recognize these co-workers throughout, but should mention in particular Robin Turner and Mike Blades, who are my partners in the Raman spectroscopy area. And then Felix Trampler is the Sonosep leader of the acoustic device that I mentioned. Okay, so thank you very much. I'd be pleased to answer any questions.
Thank you so much, Jamie. That was a wonderful talk. Especially, I especially appreciate the attention to the cell therapies around insulin production and for type 1 diabetes, which certainly strikes a chord with me in my background. We do have a really good question from a participant that I'd like to focus on first. And then, of course, got quite a few questions as well. Elisa asks if Raman spectroscopy is used only for within the cell type of imaging work, or if you're able to measure media as well. That's a really good question.
So I should have mentioned the Raman spectroscopy is used to analyze media in biotechnology. Kaiser Optical was the lead company who developed that, but there are several alternatives now. And so what they're capable of doing is analyzing or inferring the concentration of glucose and other very high concentration substances. They can assess the concentration of cells, viable cells to some extent. And so, yes, it can be used to analyze the medium, and thereby, one thing that's important to me is that means already in a similar setting, these kinds of devices are being used in the manufacture of therapeutics. And so that lowers the bar to a considerable degree for the cell therapy application, although we have new technology challenges to actually make that happen. But I think lots of good ideas on how to do it right.
So maybe you can, just so we can, a lot of us haven't actually been able to witness Raman spectroscopy in action, to visualize the process, what it looks like. Is there a traditional manner? I mean, it seemed as though from the schematic you showed, we're looking at a microscope, sort of a fixed, I wouldn't say a fixed sample, but a sample in a fixed state, if you will, though fixative. Yep. Very static, I guess I'm trying to say, instead of dynamic. Are there various modes with which you can apply this?
Yes. So first of all, it can be applied to live cells. Working with fixed cells just makes it simpler to do from an experimentalist point of view. And we're going to have a publication related to that. And so the probes that are used in bioreactors are just probes, essentially. They're just inserted into the bioreactor, and so it's non-invasive to that extent. And then the spatially offset Raman spectroscopy that I mentioned is something that can be used to actually analyze by Raman spectroscopy into a fluid through a plastic material. And so that allows it to be used and a setting that relate to culture bags. And so, yes.
That's excellent. So, you know, especially within some of the last data sets you were showing there, it really piqued my interest around the insulin spikes and the glucagon spikes that you see. And so you start to think, is there an application for this in a very dynamic setting? So as in, can you challenge the cells with glucose and watch varying levels of the peaks as you see an insulin release response?
Yes, and wouldn't that be a potency assay? So that's, of course, that's a really good point. And regrettably, only a small fraction of the insulin in beta cells is released when they are stimulated to release insulin. And so we don't expect that we would be able to discern that very easily. But we'll certainly try.
Right. So I guess that does make sense. So especially if you can apply this logic to the media, as was mentioned by Elise there, that would obviously be quite potent or quite a meaningful potency assay. I guess have you looked at any extracellular vesicle-based studies as well?
No, but I have talked to people at Kodiak, for instance, to see if I could interest them. So we'll see. Absolutely.
Very good. Yeah, no, that's obviously a field that's really exploding and getting up and interactive. I guess, you know, really those of us that have actively done the sampling and release testing and, you know, all the terminal assays where you have to pull a sample and you analyze that sample and then it's gone, right? That those cells or whatever you're studying gets effectively thrown out, is always depleting from the total number that you have to work with, to especially if you're going to deliver it to a patient. So it's a huge concern. And as you pointed out there with, even the acoustic washing steps and implementing that. If you can reduce the volumes that you're using, then, with your release testing, the reduced amount of product that you have to waste to determine your answer, the better. So this certainly has a lot of application. If
Yes.
anyone else has any further questions, please don't hesitate to populate them into the chat. But I guess one of the key questions too, Jamie, is then how difficult is it to implement these techniques into a GMP manufacturing process? So, is it equipment that takes up a large footprint? Is it something that's really relatively easy to incorporate into the process, or does it take a little bit of redesigning?
Yeah, so one would seek methods that would minimize the redesigning. And, for instance, the footprint of the acoustic systems can be designed in ways that it's much smaller. And so from the point of view of a manufacturer, that's important, especially if you're going to be running, 10 or 100 CAR T-cell therapy processes in parallel. The biggest obstacle to implementation is actually to go through the sort of regulatory approval that's required for any of these sorts of devices. And so in a normal context, it means, Thermo Fisher Scientific or whomever investing literally millions of dollars in bringing that to approval so that users can adopt them in a way that that's the biggest obstacle that has to be reduced.
Right, the chemistry and manufacturing controls. It's always the be all end all really within an application. So it bodes, it brings it to light the comparability studies that might be required and a strategy to employ these techniques from the onset of, say, your clinical trials, the early phase, and develop it throughout, right? That's very good.
Yep. Yeah, and really, what's one of the great benefits of CellCan is the possibility to work with samples from actual clinical trials in a way that we can seek correlates with clinical outcomes, or use methods that are compatible with clinical trials. And so I really benefit a lot from being involved in CellCan and really in a community of practitioners. I think for Canada as a whole to sort of bring us together and have us operate as a network makes so much sense to have the greatest chance of moving forward in this area.
It's a great point. And certainly we see a huge movement in the field for people to establish greater analytics capacity. What better way to do that than non-invasive methods, right? So that's fantastic. Well, I see our time is drawing close here. So if anyone has any last second questions, please don't hesitate to populate them in the chat. Otherwise, as our team has put up on the screen, thank you so much. If you have a chance to fill out this SurveyMonkey questionnaire, we'd really appreciate your feedback. Scanning the QR code is one of the quickest ways to get there. We'd love to hear from you and any further ideas you might have. We thank Dr. Piret tremendously. You've been certainly a pillar of our network for all these years, and we love to see the updates and what's new and what's going on out in the West Coast. So thank you so much, Jamie. Thank you to everyone for joining, and you have yourselves a fantastic day. Stay safe and stay healthy. Take care.